Hi, everyone. Welcome those of you that join us for this, the highlight of our day. We've had a very exciting day discussing engineering biology. Many of you were here before. And as I said, those that join us now, you are very welcome to be here. I am Amparo Galindo. I'm a professor of physical chemistry in the Department of Chemical Engineering. And I'm also the director of IMSE, the Institute for Molecular Science and Engineering. Before we start with the lecture today, I have a little bit of housekeeping. As you see, we are very lucky to have a British Sign Language interpreter for our lecture today. There will be, in fact, two, so there will be another one arriving. If any of you need the interpreter, please move to the front if you haven't done so yet. And because the content of our talks is quite or very technical, and some of us speak also very quickly, people like me, the interpreters will switch every 10 minutes, so you will see them switching. So yeah, I hope you're not too distracted by that. I hope especially our speaker will not be too distracted. We are very lucky, as I said, to be able to have an interpreter today. So let me briefly just introduce today's talk. Let me tell you a little bit. We are, as I was saying, the Institute for Molecular Science and Engineering. We are one of the Grand Challenge Institutes of the college. We work with a remit to facilitate and promote interdisciplinary work throughout the college. We draw on the strength of our 4 faculties, and in particular in IMSE, we are interested in problems in which molecular innovation can deliver solutions at engineering level or at function level. So that's really the work that we do. And our event today on engineering biology is a prime example of how disciplines coming together lead to better solutions. And there's been plenty of examples of that today, and we will see more in the work of our invited speaker. So as I was saying, this is our highlight event of the year. This is our 6th Dr. Theo George Wilson annual lecture. This lecture is possible thanks to a very generous award by Theo Wilson, who is a PhD alumnus of the Department of Chemistry. He graduated in 1952 here with a PhD and went on to study medicine in the US, and he now works in the US. And so as I was saying, he donated a very generous gift to IMSE in particular, and in quotes, at the time of his gift to us, he said that he wanted the money to play a part in furthering the tradition of great scientific advances by Imperial. And he said, I hope my gift will be of great benefit to students in the future. So as we made the decision of how to best use the money that he was gifting, we decided that starting this series of lectures to keep it as broad as possible and with areas of interest that can include as many of us as possible was a very good use of that money. And in addition, we created a scholarship every year to support an EMRES student working in the area of molecular engineering, because that is the EMRES that we support in the Institute. Okay, and so with this, my other task is to introduce Professor Kristala Jones Prather. We are very privileged to have her here today with us. She's the Arthur D. Little Professor of Chemical Engineering in MIT. She's also the current head of MIT Chemical Engineering. In terms of her background, she obtained her Bachelor of Science in MIT in '94 and did a PhD at the University of California, Berkeley, and graduated in '99. From her PhD, she went on to work in R&D for Merck for 4 years. So it's, again, a very interesting bio, moving from academia to industry and back into academia, which again, very, very successful, and showing how taking, or experiencing different environments can lead to very successful careers. In terms of her work, I got some information from her webpage. So she describes her work as engineering microbes to produce chemical compounds. And I think we're going to hear some of that today. And she's interested in metabolic engineering, biochemical engineering, bioprocess engineering, and synthetic biology. Kristala, as I was saying, we're very honored to have her here. She has a long list of awards. I selected a few. There were pages. So I have just highlighted some of maybe the most impressive one, also the recent ones, but I leave them there for you to read. I'm not going to read all of them. What is, I think, exemplary of the career that Kristala has is that there's there a list of awards that go from technical awards, like the AICHE award at the top, to also plenty of teaching awards, and also advocacy awards. I know that you're a big advocate for inclusion at work, for equity, which is something that is very strong for us in IMSE in particular and in college in general. So, really very happy to have you here, as I was saying. And so with this, Kristala, I'm going to give you the floor. Thank you so much for being here. She's going to tell us about building microbial factories, design, assembly, and engineering of biological routes to chemical compounds. Please welcome Kristala with me.
Well, good afternoon, everyone. It is a pleasure to be here. I have to give a warning up front, which is that my task was to present a talk that would be accessible to a wide audience. And so I did what many academics do, which is choose to talk about a lot of stuff. With the hopes being that if you don't understand the first part, maybe you'll understand the second part. If you don't understand the second part, maybe you'll understand the third part. If you understand none of it, maybe I'll tell a joke or two that you'll enjoy along the way. And if you still don't like my jokes, hopefully you'll enjoy the reception afterwards. So I do all the work in our lab is really focused on this idea of engineering microbial systems for chemical compounds. But we're motivated to do this with a very specific purpose. And I use this graphic as an illustration of how we think about where the material comes from. I'm a chemical engineering by training, and I often tell chemical engineers when I'm teaching them in our introductory class that chemical engineering is about stuff. It's about making stuff, it's about characterizing stuff. It's really about working with materials and molecules all around us. And so a natural question for me then is, where does that stuff come from? And the reality is that a large amount of the stuff that we need and that we use comes from petroleum, comes from non-renewable sources. As a chemical engineer, I'm also a person who teaches in this intro class about conservation of mass. And so if we believe that fossil fuels are actually the byproducts of fossils, then it can only be that we are consuming them at much faster rates than we're producing them. So we have a fundamental problem here. Now, the part of this picture in terms of where materials or stuff comes from that we're most accustomed to thinking about is the largest fraction. And you can see the numbers that are there are in gallons. Most of it is about energy. And there are lots of ways that we can think about displacing that mass for energy. And the example that's shown here is that of an electric vehicle. So when we think about taking the carbon that's in a barrel of oil and asking how do we power mobility or how do we power devices, there are many ways that we can think of to address that don't require the utilization of carbon. However, there's a smaller fraction of this, but a fraction that, from my perspective, is just as important. That's not about the carbon mass being used for energy, but the mass being used for mass. That is, it's about making the stuff. And the graphic that's shown there is an indication of all of the different materials, representations, I should say, of the different materials and the different places in which this carbon that comes from petroleum or from fossil materials is used for things that are very important to us. It includes the plastic in this clicker in my hand, the materials of the chair that cover the chairs that you're sitting in as well. So when we ask ourselves, how do we actually think about replacing that stuff? And we think about where does this come from? We start from crude oil going into fuels and also into chemicals. And then ask, well, if we can't keep relying upon this resource that's already there, where do we get access to carbon now, mass for mass purposes, that we could have on a regular basis, on a sustainable, renewable basis? And that leads us to biomass. So the title of the slide that you can see here is describing a paradigm shift. I've borrowed this from one of my colleagues at MIT who talks about the need to go from being based on hydrocarbons to carbohydrates, or in other words, from being based on fossil materials to being based on renewable biomass. What that means is that we need a way to think about how do we take those materials now, this biomass, and can convert that It could be converted into fuels, and especially these days, the focus on aviation fuel. I've gone on record as saying I will not fly a battery-powered airplane to the UK from the US, right? So that's an area where we have to think about, because of the density there, about replacing mass with mass. But it really is this much wider array of biochemicals and biomaterials that's attracted our attention and motivates the work that we do. Now, as a chemical engineer, this was the view I was given of a biological cell when I was a student about 35 years ago, which is, it's this thing, don't worry about it, it's just a cell. What we know, of course, is that a cell is comprised of many different reactions and many different metabolites. And I liked very much earlier today, Mark Horwath said, biology is the search for chemistry that works. Okay? And in effect, biology is chemistry. It's a collection of all of these reactions that give us form and function. And we've been able to show that you can take simple inputs like sugar that can come from biomass. In some cases, you need to provide oxygen as well, and other nutrients, those components that give us biomass. And from that, we have a rich history of producing, especially pharmaceuticals. So being able to have natural products that have therapeutic functions to be able to address different diseases. And you can see the representative molecules that are shown there, as well as the diseases they were indicated to treat. But we've also been able to use biology to produce things like amino acids, which are natural products in biological systems, and also to produce different kinds of organic acids, some of which are natural to biological systems and some of which are not. So this tells us that biology already gives us a tremendous reservoir or a tremendous set of molecules that we can choose from, but more importantly, a tremendous set of reactions that we can choose from in order to build new ways of using biology to build chemical compounds. So if we go back to the same diagram I had before, what we're really doing is taking a very simple molecule, which shown here is glucose, and we have to traverse this very complicated network in order to make a particular compound of interest. And I'll come back to this compound that's shown on the slide in a minute. As we do this, one thing we're concerned about are byproducts. In other words, what are the compounds that are made that we would prefer that the cell not make? Well, one of the things that we know is if the cell is making this compound, it must have come from a reaction in that cell. The reactions are almost all mediated by proteins, specialized proteins called enzymes. Those proteins are encoded by DNA. And so if I know where the unwanted compounds are coming from, and I can identify the protein that makes that unwanted compound, I can remove it. And we heard many talks today about ways in which you could remove them, including ways if you want to string too many things together, you may run into trouble constructing these systems to be able to do that. And this is very classic metabolic engineering. Both Kyler and Karen's talks this morning talked about pathway engineering. But one of the challenges that we've asked is what happens when what you're competing against is not some other compound that's being made, but rather it's the desire or the need of the cell itself to grow. In this case, you don't have the choice to simply knock out growth or eliminate growth, because if the cells don't grow, they don't make these proteins, the specialized proteins that are enzymes, that you need to do the chemical reactions to make the compound that you want. So we're faced with the particular challenge here, and how do we actually address our desire to funnel or to channel as much of the carbon that we provide into the specific compound that we want, while balancing the fact that the cells don't really care what we want, right? So we're working against their interests at all times. So with that as context, there are two primary objectives we've had for nearly all the projects we've had in my lab over the past 20 years. The first is to be able to increase what I'm referring to as the nodes in this network. So in this diagram that's shown here, each of these dots represents a chemical compound, so that's some molecule present in the cell, and then the lines between them represent chemical reactions. So when I say increasing the nodes or increasing those dots, what we're trying to do is to find more ways for biology to make chemical compounds, and in particular to make compounds that are specified by us for particular uses. And then the second thing that we focus on is how do we actually get enough of those compounds being produced to be significant, to be relevant, to be something that would actually be useful. So we have one part of our lab which focuses on how do we actually get new molecules, and then very quickly those projects turn into ways to try to understand how do we push the capacity of biology to do more and more of the chemistry that we want. So the rest of my talk, I'm going to walk through a series of examples of different targets that we've selected, what our particular challenges are, and hopefully you'll see a narrative that goes through in terms of general themes and approaches that we have to tackle this general problem. Okay, so I am going to start with the production of a compound called glucaric acid. There's a reference that's shown here on the bottom of the slide, and so you can see that we first started working on this a long time ago, but it has presented a number of challenges, and I always tell my students, challenges are just opportunities. So when you are struggling, remember, you have been given an opportunity to overcome something very difficult. I won't spend a lot of time talking about how we developed this pathway because, again, we generated it some time ago. But I'll just say we actually started from the end and worked our way forward, meaning we started by asking where does a compound like glucaric acid come from? It turns out this is a natural product, and we identified a bacterial source of a protein that could make that compound from a substrate called glucuronic acid. If we asked where glucuronic acid came from, we identified a mouse version of a protein to make glucuronic acid. And if we asked where we could get myoinositol, the precursor for glucuronic acid, from, there had been a study already published that had shown that that compound could come from only adding one gene to E. coli, and that was the gene encoding a protein that came from Saccharomyces yeast. So this was, in concept, a very simple pathway. It has 4 reaction steps, but it only requires 3 new proteins to be added to the cell. But the challenge that we have is that the first intermediate in this pathway is a compound called glucose 6-phosphate. How many of you have taken a biochemistry class? How many of you are getting warm, fuzzy feelings thinking about biochemistry? There were a lot fewer hands with the second question than the first one, okay? But biochemistry can be useful. And those of you who are thinking fondly about biochemistry are immediately going, yes, I see the problem. Glucose 6-phosphate is the first committed step for glucose metabolism in E. coli. Meaning what the cell really wants to do is take that glucose 6-phosphate and use it for its own purposes and not to make this compound glucaric acid that we're interested in. And just very briefly, here's an overview of the different reaction steps that happen in E. coli when it, meaning when the cell, wants to use glucose to make more of itself. And what you'll see here is that we have the primary glycolytic pathway, we have the pentose phosphate pathway, there's a minor reaction to make a small amount of glucose 1-phosphate. I'm not going to go into a lot of details here, I'm just going to say that our goal is to be able to take glucose 6-phosphate and have more of that be available to make the compound that we're interested in. We use both what was available in the literature and some of our own modeling work to identify the ZWIF protein, or glucose 6-phosphate dehydrogenase, as something that we could eliminate from the cell. That way now we know that the glucose is mostly going down that pathway shown in the middle. And then we also identified one particular enzyme in the middle as the point where if we could control the ability of glucose to go through that particular point, we could regulate whether or not it was being used for the cell to grow. And you can see where biomass is indicated on the slide as something as indicated of the cell growing, or to be able to have what we call flux, or essentially have flow through that point be restricted such that we have more of that substrate available. And if we have more of it available, we have more of our first intermediate and ultimately more of our glucaric acid being produced. So conceptually, we knew what it was that we wanted to do, and then the question was how? What is the mechanism? What are the specific pieces of proteins or DNA that we're going to assemble together into a cell to be able to build what we call a metabolite valve? We call them valves because if you have ever turned on a water faucet, that faucet has a valve in it that allows, when you open it, for the water to flow, and when you close it, for the water to not flow. In our case, we want a valve that when it is open, it allows the glucose to be used for growth, And when we close it, and we don't close it all the way, but when we close it just a little bit, there's less of it available for growth and more of it is available to go into the pathway that we're interested in. So we actually demonstrated a couple of different ways of controlling this. I'm going to give you an example of one that has been a theme we've carried throughout other work in the lab, and that is using something called quorum sensing. And I'm going to pause and go through this in a little bit of detail because if you've heard of quorum sensing, it makes perfect sense and you don't need any explanation at all. If you haven't heard of quorum sensing, it makes no sense. And I can give you a little bit of a foundation to help understand what it is that we're trying to do. So I'll turn to this one to make it a little bit easier. Our quorum sensing system has a couple of key features. One of them is a special protein, a regulatory protein. You can see it here as ISA-R, whose job it is to sit on DNA. And it will bind to a specific sequence of DNA and will sit there. A second component is what we call a ligand or a signaling molecule, which is a small molecule that will interact with that regulatory protein. And when the molecule is absent, the protein sits on the DNA. When the molecule is present, past a certain point, the interaction of that molecule with the protein causes it to come off of the DNA. In this particular case, we need the DNA to be open in order for us to make a protein. So DNA goes to RNA, RNA goes to protein. So if I don't have any of this signaling molecule, my regulatory protein sits on the DNA and it prevents me from making the protein of interest. If, however, I have enough of this ligand accumulate, then the protein comes off of the DNA. That allows then for another specialized protein called an RNA polymerase to come in and convert the DNA into RNA, and that allows me to have protein. Now, the last component I'll mention here, so I've talked about the places where the regulator binds. Those are our promoters. The regulatory protein I've mentioned. This last component is called an AHL synthase, and the AHL is the signaling molecule or the ligand, that is the small molecule that binds to the regulatory protein. There's another protein in the cell that produces that molecule. And so with those key components, what you have now is a cell that will produce the signaling molecule. At low amounts, it leaves the cell and it just floats around in the space where the cell is being cultivated. When it reaches a certain concentration, it will come back into the cell, and when it comes back in, it now is able to interact with this regulatory protein in a way that it can cause it to be displaced. Okay, so why is it called quorum sensing? Well, typically we think of a quorum as being a sufficient number of people or sufficient number of items for some action to take place. If I now have this signaling molecule being made by the cells, the more cells I have, the more quickly I will make that signaling molecule. And there's some critical amount of the cells that corresponds to a critical amount of that signaling molecule to trigger the reactions that we see. Hence, if the cells establish a quorum, then they're able to trigger gene expression. The benefit of this to us is that it allows us to construct a system where what we're actually doing is allowing the cells, based on how quickly they grow and how quickly they make this signaling molecule, decide when they want to switch our valve. This diagram, the way I've shown it, has the cells starting where the protein to be controlled is off and is turned on. But the particular system that we use, and the reference for that is shown here, and it's named here, actually has a different mode where that same regulatory protein can bind to a different sequence of DNA located a little bit further ahead of where the first site is. And now when it binds, it actually brings with it the protein that we need in order to make our target. The same interaction happens where as that quorum sensing molecule is built up, the regulatory protein is displaced, but this time when it comes off of the DNA, it takes the RNA polymerase with it. So now we have a configuration where we start on and we're turned off. So this works great for the case I described, where we want to start by letting the cells grow to make all the proteins we need to trigger our reactions. And then at a certain point, we want to say, you've grown enough, you've been selfish enough with our carbon, and we're going to have you stop growing and then utilize the carbon that we're providing in order to now have our product of interest being produced. So we can vary the promoter to go from on to off, and then we don't know exactly when is the right time for switching, but we can actually build a library that expresses or makes that signaling molecule at different rates, and then just screen the library for the best time for the cells to switch. Okay, so that's an overview of quorum sensing, but I want you to take something else from this slide, and that is that in general, we can control whether or not a protein is being produced by being able to control whether or not a regulatory protein is sitting on that DNA or coming off of the DNA. And if we have multiple choices for how a regulatory protein can be displaced from the DNA, then we have multiple ways that we can control the flow of metabolite and therefore control the production of compounds. Okay, so I'm just going to show one result of having gone through this whole process of now looking for the right point to be able to switch between producing cell mass and producing a product of interest. And in this particular case, we actually wanted to make sure that the system that we developed would work at different scales. So we had a panel discussion right before this talk where one of the questions that was asked was about translation and how do we actually consider translation and going from academic research into what's happening in a company. And it's something that we think about a lot as well. So we want to make sure when we're building new systems, new tools, that we've tested them, not just in the smaller scale systems that are useful for being in a research lab, but also in these bioreactors, which will give us a much better indication of how it might perform if we go into manufacturing. So the standard that we use is actually a shake flask. So that's a 50 milliliter culture volume and a 250 milliliter vessel. There are 4 different samples that are shown here on this chart. To the far right is a wild type. So this is a cell that doesn't switch at all. It has the pathway, meaning it has the genes that it needs to make our product of interest. But you can actually see under these circumstances for these cells, they don't make the product. The product is shown in the blue bars here as glucaric acid, and the yellow bars are one of those byproducts. And we had actually already tried what the typical method is, which is to knock out the genes encoding, in this case for acetate, but that gave us a cell that wouldn't grow at all. Okay? So we were actually faced directly with this problem I presented, which is what if you can't knock out the pathway for the thing that is your undesired byproduct? But in this case, it was both growth and an unwanted acetate molecule. The 3 other strains that are shown here are 3 different strains that will switch at different times based on the rates at which they are making this signaling molecule. And so we have something that switches very late, so it looks a lot like wild type. It makes no product of interest, only acetate. We have one that switches pretty early, in which case it makes some product, but it can't quite figure out what to do, and it makes a lot of acetate. And then we have what's our Goldilocks strain, which switches just at the right time, and it accumulates no acetate, and it makes only product of interest. We could test this on a scaled down model. I actually saw one of these at White City yesterday called a biolector, where you're using 1 milliliter culture now, so 50-fold smaller than the scale in the center. And you can see that both qualitatively and quantitatively, we're achieving about the same amount of product being produced. And then the next step was to look at how this might scale up into a larger reactor. Okay, how many PhD students are in the room? Okay, I'm going to tell you a story, and you can't tell your advisors. Okay. So at this point, the student who was working on this project came to me and said, I don't want to do the experiment. I think that's a phrase that has never been spoken by any of you who raised your hands. It's a unique phenomenon in the United States. And then I said, so tell me why you don't want to do the experiment. And he said, well, in these strains here that are making a lot of acetate, I think that's causing the pH to go down. Yeah, it's an acid, right? You make a lot of acid, the pH is going to go down. And then he says, well, I think it's the low pH that's actually causing the cells to not be able to make product, because we know that if the pH is too low, you actually have trouble transporting glucose into the cell. Okay. And then he says, well, if we're in a bioreactor, we can control the pH. And if we control the pH, I don't think the system is going to perform quite as well as it otherwise would. Therefore, I don't want to do the experiment. So you have a conversation that goes like this. There's no such thing as good data or bad data. Your reaction to the data might be good. Your reaction to the data might be bad, right? But we're here as a search for truth, right? In other words, go do the experiment. Okay? And he was kind of right. So what happened when we had the large-scale reactor is that the wild-type strain, so this is shown here, did make a lot of acetate. But E. coli can reassimilate acetate. It can take it back up. And with the pH being neutralized, that's exactly what it did. It made a lot of acetate, then it took that acetate right back up, and then it could make product. However, this actually had a much lower titer than the case where we were controlling it much more directly. So we had more biomass, less product in that case. In the case where we had our valve, we accumulated no acetate, less biomass, more product. So the key take home here, one is that we actually were able to show the consistency across scales, which is very nice. And then the other, I borrowed this line from my good friend Tony Sinsky at MIT, who says the best metabolic engineering projects are ones where you start from zero and make anything, because now you get to divide by zero and say that you have infinitely improved the performance of the system. Okay, that may be a stretch, but what it really showed us is that we took a case where, for this particular environment, for these strains, we had trouble producing a compound, and then our ability to actually regulate the flux of glucose to keep it from going into endogenous metabolism kept us from accumulating a lot of acetate and allowed us to make our product of interest. Now, I mentioned that this was a pathway we've worked on for a long time because it's given us a number of challenges, and those challenges are opportunities. And I want to tell you very briefly about a different step in the pathway, and this is shown here where we use this mouse protein to go from a compound called myoinositol to glucuronic acid. And we knew from our first publication that the activity of this protein was very odd. It was much higher if you synthesized the protein when its substrate was present. We also found that the activity was highest early on, and it dropped off much more rapidly than the other enzymes in the pathway. It's not unusual for an enzyme to be highly active initially, and then after a few days, the activity to go away. But this one, the activity went away very, very quickly. So that actually gave us an idea that if we could delay the expression of that enzyme until its substrate was present, then we would perhaps see higher titers. And I'm showing here a graph where we did that experiment, where we had two different enzymes, or two different genes. We controlled then the expression of those two different genes to make the enzymes with two different inducers. And if we delayed expression of the second one relative to the first one, you can see we got higher products being produced. So that was nice, but again, if we're thinking more towards translation, we don't want to have an operator go into the system and decide, now I want to induce this second protein. Instead, what we would like is for the cells to be able to say, okay, I need to be able to turn on expression of this protein now because this molecule that I need to be present is there. So if you remember when I described quorum sensing to you, I said the key features were a protein that binds to the DNA, and then something else, in that case it was this AHL, that when it is there, allows for the protein to come off of the DNA. Well, it turns out there is something exactly like that that responds to the substrate for this reaction, which is myoinositol. It's a protein found in a completely different microbe than E. coli. It's in an organism called Corynebacterium glutamicum, and it works exactly like the system of quorum sensing works, where if the molecule it responds to is not present, it sits on the DNA, and it sits at a spot that we choose, and we chose a spot so that it would turn off gene expression. And then when now the product of interest is there, what we're able to do is to have that myoinositol give us the ability to turn on expression of that gene. These we now call biosensors. Okay? And just roughly speaking, a biosensor is anything that will respond to a particular molecule. In this case, they are biological molecules, they are proteins, and they're responding to metabolites of interest. And so what we wanted to do was to build then a biosensor that will allow the cell to decide when is the right time to turn on this particular protein. I just want to show you how we build these biosensors. I'm going to show you several sensors coming up later where I'm not going to talk about how we build them. But one key feature here is that we know if this protein sitting on the DNA has to be displaced by this new molecule coming in, this ligand, then how much of that ligand we need to displace it is going to depend upon how much of the protein we have. So what I'm showing here in these 5 different colors are 5 different samples where the regulatory protein is being expressed either at very high levels, that's the lowest, or at lower levels, which is the one at the top. And the line that's fairly straight across the top is the case where we're not regulating anything. So we just have, in this case, a GFP reporter being turned on all the time. So you can see if we don't make a whole lot of that regulatory protein, then it doesn't take a lot of the ligand being added before it can give maximal expression. But if we have too much of that protein, we actually can't add enough of the ligand in order to have it be displaced. Okay? But in all cases, what you see is that we are getting expression of this protein as we add more of the myoinositol, and we can sort of tune how much we make based on how much of that ligand is present. That experiment was done if we just added the myoinositol to the system. We now want to see what happens if we're making it inside the cell, because that's the use case that we're interested in. And what we see here, first the sensor responds in a dose-dependent manner. If we're now looking at what happens when we're making this compound in the cell or not making it in the cell, you see the same sort of reaction, that where the lowest activity is there of the promoter, we get the highest signal from making the compound inside the cell, and where we have more of it, we have less. The next part then is to actually use this system to directly control the expression of the production, rather, the compound that we're interested in. The no IPSA case is the case where we can't turn it off and it's on all the time. And then we have these 5 different cases, and we don't know exactly when is the right time for gene expression to be triggered, but we can let the cells choose. And you can see there's one case where we actually have this expression being controlled by myoinositol, and it results in an increased expression of increased production of the compound that we're trying to make. So the last slide that I'll show is that the first case I told you, or showed you rather, of being able to control the amount of glucose going through the cell. In this particular case, even though they're on the same pathway, they have 2 totally different mechanisms. So we can put them together, and what was before a 2 to 3-fold increase in improvement for either one of them by themselves actually became about a 10-fold improvement when both of them were put together. We have used this general approach of saying, okay, how do we control the flow of material, both to continue to look at new quorum sensing systems. I'm not going to talk about that today, but we also have been inspired to think about new ways of using biosensors, that is, using ways for pathways to be controlled based on particular small molecules in a number of different applications. And so I want to introduce, I forgot about this slide. I told you it was a new talk. Okay, so let me come back to glucaric acid and this compound that we're producing. And I want to tell you why we're making it. We originally chose it because it was on a list of compounds that, from a report of the US Department of Energy, there was a similar report that came out from the EU a few years after that. The first report came out in 2004. The next one came out around 2010 that identified this molecule, glucaric acid, as an interesting compound. And one of the things that they pointed to had to do with the efficiency of cooling towers. Okay? Now, they didn't talk specifically about cooling towers, but what they talked about was water treatment and water usage. And in fact, what was known was that phosphates were the primary molecule that you would add to water treatment products to be able to chelate heavy metals in industry. The problem with phosphate is that if you have runoff into natural waterways that are normally phosphate limited, algae that exists in those water bodies will now see a lot of phosphate. They have plenty of sunshine. They will grow. As they grow, they block out the light for the aquatic wildlife underneath. So you have a process known as eutrophication, or essentially as an algal bloom, and that actually will kill the fish that are underneath it. Well, what we knew from that study and have since been able to show, and this is Callion as a startup I'm a co-founder of, what we've been able to show working with partners there is that glucuric acid is a great replacement for phosphates. Again, that was actually known for the report. What we didn't know until recently is that actually using glucuric acid in place of phosphate for the water that's treated and goes back into these cooling towers actually cuts down on fouling. So the chemical engineers in the room are now thinking, yes, this is my thing. She's talking about heat exchangers and cooling towers. I'm still a chemical engineer at heart. The biologists have no idea what I'm talking about, by the way. So if you actually reduce the fouling, and fouling is essentially a process where the inner lining of the pipes have metals that deposit on them. What that fouling does is actually reduce the efficiency of the cooling towers. And so we've actually done some studies, or some back of the envelope calculations, I should say, based on some initial studies that show that not only can you have cleaner runoff if you replace phosphate with glucuric acid, but you can actually reduce CO2 emissions from industrial waste facilities by the equivalent of about 230 million metric tons per year. And that can result in cost savings of about $60 billion US to the need to be able to refurbish or to renovate or get those facilities back up again and cleared of all the fouling. So I had promised I would tell you what glucuric acid was used for, then I forgot that I had promised I would tell you that, and then the slide was there to remind you of it. So this is something that we're pushing forward with in partnerships to be able to bring cleaner solutions to be able to address the environmental demands for industries that we can't quite get rid of yet, but need to be much more efficient in their carbon footprint. Okay, now I'm going to continue with the biosensor study, and there's a lot of details on these slides. In the interest of time, I'm not going to go through them in a lot of detail, but for those of you who are working in this space, if you want to ask me questions about it afterwards, I'm happy to go into a lot of detail. In this particular case, what we decided is that we wanted a biosensor to be used to help us control whether or not a pathway to make a particular compound was turned on or off. And again, I'm going to wait and give you the punchline for why we wanted to do this a little bit later. But I'll say in this case, we were interested in a very specific substrate or a very specific starting compound, which is called galacturonate or galacturonic acid. The substrate was actually more important to us than the product in this particular case. So based on the substrate that we had available, we actually started to look for what some interesting end products might be that would be useful to make from galacturonic acid. And we settled upon glycerate as a test study for that. And you can see to the far right, there's a reference that describes all the ways in which glycerate can be useful as an industrial chemical. Now, it turns out that E. coli cells have a way to use galacturonic acid, but in particular, if we introduced two new enzymes into the pathway, we could produce this intermediate that was already known in E. coli metabolism, and then the gray arrows here represent what the cells will already do. So if we introduce two steps, we could then turn the rest of it over to biology. Biology is a search for chemistry that works, right? We could use the chemistry that was already shown to work in E. coli to make this glycerate. So we developed a sensor for this in the same way that I described before. I'm showing here what those traces look like, but those are just different ways, as I had said previously, of having different strings of promoters. In this case, we also have different places where we've put the strings of DNA that the regulator binds to. But all these different versions give us a range of different constructs that we can use. And in this case, rather than letting the cell decide, we decided we wanted something that had a very high dynamic range. That means that if we take the maximum output and divide it by the minimum output, then we have a high change there. And we wanted a low leakiness, meaning that when our substrate is absent, we want the expression to be as low as possible. So I've highlighted there the particular construct that we chose. And in this case, it became very important that we find a way to control the pathway to make glycerate. And that's shown in this particular slide. So this is actually a set of growth curves that shows how the cells grow in the process of making this product of interest. And I want to point out on the far right, this gray line. That gray line is the cells growing if we're trying to make this pathway all the time, meaning it's always on, we're not controlling it at all. Those are very unhappy cells. Okay, so this is the growth as a function of time. It takes them a lot longer before they start growing. They eventually do get to the growth of the other strains, but they are not very happy. If, on the other hand, we let the cells grow without turning the genes on, and then we induce them, then the cells grow like this. And all these different traces show what happens if those genes are being turned on because we've added galacturonic acid, meaning we're using our sensor to do the work, or if we've added something completely separate, just a regular inducer you can use for almost anything called IPTG, and that's shown here as IPTG. So those cells are equally happy if we induce with something that's not associated with the pathway than they are if we induce with something that is associated with the pathway. And in fact, they actually produce the same amount of material if we are inducing with the substrate that's already going to be there, or if we're inducing with something else that we have to add. So what I'm showing here, this is now the substrate being consumed, this is the product being produced, and the peak bars for both substrate consumption and product formation are in the case where I have this other molecule, the IPTG, and the dark blue bars are what happens in the case where I let the cell decide to turn on the pathway when that substrate is present. As a note, these gray cells, which didn't grow very well, they also cannot consume much in the way of substrate, and they cannot make much in the way of product. Okay? So what we have here is a system where we know that inducible or controlled expression is required, and we've been able to show that we don't have to spend the money for an extra inducer. We can just let the cells respond to what we're already adding into the system and go from there. Okay, here's why we were interested in this. We were interested because we had a project from the US Army where we were presented with the following challenge. Army bases have a lot of food waste, okay? And the amount of food waste that you have is quite variable. And we all know about composting, and you can make biogas from composting, and composting works with pretty much anything. But the Army was interested in whether or not you could take a really heterogeneous mixture like this and use it to make something that wasn't a gas. Could you make a specific chemical compound that, for example, might go into a fuel cell and it could make electricity instead of making something else? The problem with that is this food waste is going to vary a lot. It's going to vary based on what was cooked one day versus the other. It may vary based on how good the cook was one day versus the other and how much people actually consume. But our challenge is, how do we take this very complicated mixture and get a very specific product that comes out of the end of it? Okay? And as you might imagine now, since I said I would tell you why galacturonate was something that we had chosen, when we said to the Army, okay, we want to take this challenge on, what are the different carbohydrates present in food waste? Galacturonic acid was very high on the list. It turns out it's found in fruit peels. And then there was several other compounds that were high on the list as well. So our basic challenge that we had here was, could we actually engineer a system that would have stable, consistent output, even as the input was varying all over the place? Okay? So to do this, what we decided is that we needed sensors. And we needed sensors that would respond to the different things that might be in food waste, to only turn on a pathway to utilize one particular compound when its substrate was present, and leave everything off. And if both substrates were present, turn both of them on. So just very briefly, on the left-hand side is the sensor we already built for galacturonic acid. We use the exact same process to build one for gluconic acid, which was another of these compounds on the list. And what I'm showing in the chart here is just the fact that the galacturonic acid sensor responds only to gal and not to gluconic acid, and vice versa. The gluconic acid sensor responds only to gluconic acid and not to galacturonic acid. We actually then decided to build pathways from each of these compounds to glycerate. And we needed a different pathway, and I'll show you in a couple slides why we needed a different pathway. But we still are relying upon biology doing what it already does, which is that in this case, we're letting the cells utilize known and endogenous pathways to make a particular intermediate, and then we have to add 2 new proteins to get to glycerate. I'm going to skip through the next part fast, both because I'm running out of time and because it's only going to be comprehensible to a few of you, but because we talked about the need to knock out different genes. I'm showing you the results of that exercise, which is knocking out different versions of all these other ways that those molecules can be used. And you can see doing that gives us a lot of cases. We picked the best performer in that case. We also can do some other common tricks in metabolic engineering, which involves looking at how strongly the genes are being expressed by playing around with ribosome binding sites. That gives us some improved performance. And then we can also, because we have 2 genes now, play around with the order of those 2 genes, and that actually leads to the highest productivity that we could see. The reason we had a different pathway is that now I'm showing you adding gluconate to the system, and I only need 1 additional enzyme in order to make now the same compound from 2 different products. I'm not going to show you all the details of that. I'm just going to say that this pathway is now regulated based on gluconate presence using this gluconate biosensor. We did a similar sort of exercise to identify what's the best activity we can find from there. And in this case now, what we did was to start with a system where we had each pathway individually. That's the data that's shown here. We have almost 2.5 grams per liter from galacturonic acid, a little bit more than 1 gram per liter from gluconic acid. And then we put both of the pathways in the same cell, but then provided only one or the other of the 2 substrates. And something very interesting happened. We actually got less production from galacturonic acid, and we got more production from gluconic acid, but they actually started to even out. And we have no idea why. And then as we tried a different mixture of the 2 substrates, we actually found that they all pretty much evened out to the same amount. So we started with this objective of could we take multiple substrates and then engineer them to be consistent when the input was variable. And it turns out it did that on its own. We got essentially consistent output irrespective of the input would be. We've tried a number of ways to explain why this happens, and again, we really don't know. I am out of time, so I'm going to skip through the last example, and I'm just going to point out one thing we've been working on, which is to deal with plastics use. We had a talk about plastics earlier today from Jose Jimenez, and so I'm not going to repeat all of this. The bottom line is that we know plastics are a problem, and that accumulation of large amounts of plastics in the environment is pretty damaging. But what we also know is that we need plastics for a lot of the function that we have in our everyday lives. It makes our lives better. In some ways it helps to preserve food and make it more available. In some ways it helps to actually provide important medical devices. So what we've been working on with collaborators is how can we think about designing plastics that are built for their end use, or essentially built to be degraded. We've thought about this from the whole cycle. It starts with, can we actually identify monomers that we can produce biologically that can go into those plastics? I don't have time to go over this, but I will just say we have worked on this and have looked at being able to develop pathways to new kinds of monomers that will give us the functionality that we need in biologically accessible polymers without having to go through the traditional ways of using petroleum in order to produce them. And we actually rely upon nature for this as well. So we were inspired by a known pathway for degrading amino acids, but we decided rather than degrading those amino acids, we would be able to produce the precursors for them to make these compounds of interest. And so we were able to produce 2 new compounds. This work has not been published yet. It's currently under review. The titers are very low, and so now we have to switch into that mode I talked about before, which is how do we get more of something once we've shown that we can make it. And then the last part, this also sort of harkens back to a talk earlier today, is how to actually prove that these polymers are biodegradable. The well-known assays for doing this rely on single species, and they take a long time to do. So we're working with collaborators as well to develop high throughput ways to dramatically reduce the screening time so that we know we can get the materials that we need. We know they can be produced from resources that are more sustainable and environmentally friendly. And when we're done using them, we don't have to worry about them persisting in the environment indefinitely. I'm going to skip over the summary and just say biology is great. And acknowledge the folks who've done the work. Their names are highlighted here, and they were on the slides as we went throughout. I'm delighted to be here. I thank you for your attention and for your laughs at my jokes, and I'd be delighted to take any questions.
Thank you, Kristala, for a wonderful, absolutely wonderful talk. Yes, I'm happy to take a few questions.
It's late, but don't be shy.
Yes,
2 minutes.
Can I ask you to introduce yourselves as you ask the questions? It's easier for the speaker to know where the question is coming from.
Yeah, my name is Nick James. I'm an electrical engineer. I'm just curious, you mentioned plastics and you say they're needed in our lives. Can we move more, especially with food containers, to metal containers which are not oil-based, or maybe to glass? Milk bottles used to be in glass, not plastic. We were using, they work perfectly well, and they're recyclable much more easily.
Yeah, so it's a great question. So in this case, I'm talking less about, when I say food, less about containers for just transporting in the short term, but more about packaging for being from the point of harvest to preparation to the market. So what has happened over the years is that the shelf life, especially of produce, has increased dramatically as a result of specialized plastics and materials that have these barrier properties that prevent oxygen. So when I was a kid, you could not buy salads, lettuces that were already cut up, because as soon as you cut them up and put them into a package, the exposure to oxygen would cause them to rapidly degrade. So the fact that you can go into the supermarket and find rows of all different types of lettuces, and you can pick them up and they've been on the shelf for a couple of weeks, it's because of the specialized packaging that's there. So that's one of the examples of something that's had a pretty significant impact on the quality of human life. We still have tremendous amounts of food waste, but that innovation actually resulted in a dramatic decrease in food waste, because you could transport foods, fresh foods especially, in longer distances, and you could maintain them for longer when they arrive at their end use. And glass and metal won't do that. But you're absolutely right. It's easy to convert if you're looking at only having something for a day, takeout containers, things like that. Absolutely, there are ways to do it. You still have to worry in some cases about reusability and making sure that you have reuse or that you have recyclability. Otherwise, we'll run out of metals at some point too, right? So all this ends up being about resource conservation. But the plastics for food are really about these specialized cases where we've been able to maintain the shelf life and make more foods available to more people because of the materials innovations.
Hi, Kristala. Thanks a lot for the talk. I loved it. I'm Jose, by the way. I don't think I need to introduce myself. Yeah, I just wanted to ask about the comment you made about low titers, and that it's better to produce something compared to not producing anything. So, that's the kind of argument that normally I tell to my colleagues that do scaling up, and they are typically completely unimpressed by the peaks that I saw them, the HPLC chromatograms. So how low is low, and what makes something really, I'm not going to say profitable, but something that makes sense to do for industry?
Yeah, so great question. So I would say that the part I just raced through at the end, we had 2 different compounds, one that was about 50 milligrams per liter, one that was about 700 milligrams per liter. The closer you get to in our experience, the closer you get to 1 gram per liter, the more hope you have of being able to convert that into something that, quote, matters, right? But the reality is what you said you weren't going to say is what really matters, right? So how much you need to matter depends on the value of what it is that you're trying to produce. I talked about biofuels as part of the background because I have a history of working on biofuels. But the reality is that at the time I was working on biofuels, this is being recorded, okay? I should be careful what I say. All right, so let's say today. Let's not talk about the time I was working. Today, the cost associated with biofuels is just too high, even if you are looking at environments where, in Europe, for the example, the cost of petrol is much higher than it is in the US because Europe attempts to have a tax assessment that reflects what the actual cost is, and we don't do that so much in the US. But even though petrol here is several-fold more expensive than it is in the US, it still is cheaper than what it would cost in order to make a comparable biofuel from biomass. But it almost doesn't make sense to do that. The cost is one part of it, but when there are other alternatives, given the fact that biomass, by the way, is also going to be limited, right? So there's lots of things that we're going to want to try to make from it, and the scale at which you have to replace fuels is just so high that by the time you do the scale and you do the cost, it makes sense only for those areas where you don't have any other options, but where you have other options, you explore them, right? So in a case like that, it ultimately is going to come down to what are the real costs, because the costs are going to vary based on supply and demand, and where can you actually produce enough to make a difference. There is an entirely different set of molecules that have another wide range that go from specialty chemicals to commodity chemicals. And in general, that curve is inverted, where the higher the volume, the lower the cost, okay? Those are not unrelated to each other, right? You can make a lot of something, or rather you can purchase a lot of something only if it has a low cost. And if it has a high cost, you can't purchase very much of it, so you don't make very much of it. So in part, it matters if you're looking at those very high value compounds, you can do the cost calculations and convince yourself that 20 grams per, actually, you can convince yourself incorrectly, in my opinion, that 5 grams per liter is enough, because I've heard people say that. But you can even get to 20, 25 grams per liter and actually have something that's sort of useful. But the further along that curve you go towards higher volume, then the higher your concentration has to be, because your separations cost, now I'm a chemical engineer again, right? Your separations costs are going to be inversely proportional to your concentration. So it's not that you can't make something at 20 grams per liter and sell it. It's that the cost to purify it compared to if you had 120 grams per liter is going to make your production costs so expensive that you can't do anything about it. So the markets matter is the overall message, right? In terms of how much you need in order to say, okay, I have something. There's a technical answer to that, and my technical answer is once you get to about 1 gram per liter, you have some flexibility to be able to push higher. But the ultimate answer in terms of what matters is all about the markets, whether or not what you're making has to be sold at very large volumes for very low cost, or whether you can do it for much smaller volumes at much higher cost.
Thank you. Yeah.
It's a big auditorium.
Thank you. Ellis O'Neill, University of Nottingham. It's fascinating talk, and I think we're all probably quite sold on making stuff in biology here. But when you show an oil drum at the beginning and you're looking at just that little bit we turn into useful products,
yes.
How much waste are we going to be making with this engineering biology, synthetic biology, that we're going to have to process and deal with in some way?
Yep.
And yeah, we're taking CO2, turning it into glucose, turning the glucose into something, and maybe back into CO2.
Yep.
And purifying your compounds from water as opposed to from crude oil.
Yep.
You've got thermal capacity of water. You've got a lot of stuff you're going to have to deal with at some point.
Yes. So great question. So this is the other one. When I said that people can convince themselves that 5 grams per liter is sufficient, but it's not. In those analyses, I think what's been forgotten is the cost to deal with waste. Right. So the first answer is you actually, if you're going to minimize the waste that you have, you do have to get your titers, your concentrations up high enough that you can minimize the waste that you produced, right? So if you're minimizing the waste that you produce, then that's one issue. For biological systems, though, there's 2 parts of this that make them better than comparable waste when you're dealing with oil and gas. One is that one source of waste is actually the biomass. We have precedent for this, which is in the ethanol industry. Tremendous amounts of yeast biomass waste, quote unquote, that's being produced to make ethanol. It's not actually wasted. It's dried down and it's sold as animal feed because there's an extraordinarily high in protein content. Right? So we actually have a way of reusing the solid biomass waste. There's also ways you can hydrolyze it and sort of recycle it back to the system if you need in order to deal with that. So one aspect of the waste, there are other ways that you can use it because it's non-hazardous in a way that you can't use the waste that comes from traditional petrochemical refining. And then the other is that you're right, there's a lot of water, but because it is water, and it doesn't have organic solvents in it, your ability to be able to actually rehabilitate that water is useful as well. So there are studies that are being done to try to figure out the extent to which you can do some partial cleanup and actually recycle that water such that you're not constantly having to bring fresh water in. The last point, because you made a comment about the separations processes and potential. So biological systems, I'll put it this way, we can't distill everything. Right? So you're absolutely right. If the answer is it's going to be in water and we're going to use distillation the same way that we do with ethanol, it doesn't make any sense. The energy demand is going to be too high. The costs associated with that are going to be too high in terms of the CO2 footprint. But these kinds of systems give you additional possibilities. There are new membrane separation techniques that are being developed that are much less. We don't work on this. This is just things I'm aware of. There are new membrane separation techniques that are being used. And you also have the potential for new kinds of configurations. So I'll give you one example of a project that we worked on that we're back in the biology phase of it, but in which it had gone towards more of a process engineering phase. We were working with, Tom Ellis mentioned earlier today, non-model organisms. So we were working with a non-model microbe, something that had been isolated from a natural environment that is essentially an underground CO2 reservoir. And the carbon dioxide in that environment is under high pressure, so it's in supercritical form. Supercritical CO2 is actually considered to be a sustainable solvent. Okay? And so we had a collaborator who's an environmental microbiologist who sent a student to where this CO2 mine was, pulled up what are called formation fluids, so essentially the liquids that come out of that, and after several rounds of selection, isolated this microbe that will germinate and grow in the presence of supercritical CO2, which, by the way, kills most everything else. Right? So we came up with this great idea. A lot of great academic ideas, by the way, are magical, fantastical, right? Our fantastical idea was, okay, we're going to do two-phase fermentations. We're going to use water, which is what the cells need to grow in, and then we're going to have this supercritical CO2 layer. And the benefit of that was twofold. One was nothing's going to contaminate it because supercritical CO2 is going to kill everything else. But the other is that the products we were targeting to make would phase separate into the supercritical CO2 phase. It's immiscible in water. You could actually extract it pretty easily. And all you need then is a slight depressurization. And then whatever it is that we're producing water-soluble molecules, those crash out of solution. You repressurize the CO2 and recycle it. And we did modeling there to show that you could potentially reduce the separations cost compared to distillation. I think the number was something like 75%. So all of this absolutely takes continued innovation, but at the base level, because what you're working with is predominantly water and it's non-hazardous, we have a lot more options for how to deal with the waste.
Thank you.
Okay. Quick question.
Thanks very much for your talk. Gabriel Abrahams from the University of Oxford. Also in the panel before, Tom mentioned this idea that we should be using all our thousands of cells to get lots and lots of data. And so I guess I was wondering, in your talk, there was this general theme, I think, of kind of very lovingly and carefully handcrafting you look at what you want and what you have and picking the proteins to put into your cell that will create that pathway. And so I guess I was wondering, if there's a future here where this kind of design can happen in some sort of high throughput way, or where you see that going in the future.
Yeah, so I'll attempt to answer it, and then you tell me if I didn't answer the question that you really asked. Okay. So I heard Tom say that, and I immediately thought of the place where we use that is when we were trying to do directed evolution. And I didn't talk about any of the work here, but we have some adaptive laboratory evolution experiments going on, actually in relationship to the project I just talked about with supercritical CO2. And we've also had studies where we're trying to specifically evolve individual proteins. And in those cases, you absolutely are reliant upon all the diversity. You need millions of cells because you don't know which cell is going to be the progenitor for the phenotype that you're looking for that you can grow up in large amounts. So there are certainly certain use cases where we want to take advantage of that. And there are also cases where if we had a biofoundry, right, which I got to visit yesterday, then we would have the ability to scale down in a way that you could have the high throughput that's necessary for a lot of these different cases. When we are looking at generating diversity, that's when we focus on, can you do 1,000 experiments in one vessel? When we get to the point where our objective is to try to be consistent and robust, we want to do the opposite. We actually are trying to minimize heterogeneity as much as possible. There was a talk earlier today about heterogeneity as well. And we are, at times, we want to understand it to be able to really engineer around it. But our objectives, if our focus is ultimately on commercialization and translation, in that case, in the canonical case, I should say, you actually want to minimize the diversity. Now, there's a different case or different set of cases that have yet to really emerge where I think the exact opposite of what I said is going to be true. I gave this example of looking at food waste as a starting material to make compounds from. And everything I talked about today is work we do in E. coli, and I show this example of building these pathways in E. coli. I am under no illusions that E. coli as a host could be engineered to the point where you could have a single E. coli culture, and it's going to be able to take banana waste and hamburger waste and pasta waste and convert that into anything. But I do think you could have a consortium of organisms, right, that are each designed to interact with each other, where some of them are going to be activated and others aren't, based on what the input feedstocks are, and that you could engineer that consortium in a way that you would have consistent output. And one of the reasons I believe that is because it already happens. It's called anaerobic, it's actually called enhanced biological phosphorus recovery, EBPR, is a wastewater treatment process that has been in operation for decades that uses very complex communities, one set that's anaerobic, one set that's aerobic. It switches between them to make sure that you can consume all of the organic waste that you have there. And it works until it doesn't, right? But it'll work for a long time, and then it'll have an upset, and then you sort of restart it and it works again. So I think as we think about much more complicated inputs, when we get to the point where we're trying to address the waste remediation problem on the planet, and we think more about circularity, that the complexity of the inputs is going to require us to think about how do we have more complex processes, but it also means changing how we've, and I say this as a chemical engineer, changing how we've traditionally thought about those processes, how we characterize them, and how we design them for tolerable amounts of upset, right? Because the traditional way that you design a production facility is for it to run until you shut it down. But the more complexity we have in these biological systems, the more they are going to do whatever they do, right, without us knowing it, and we're gonna have to reset. So we're gonna have to design new processes. We're gonna have to learn how to better characterize them and learn how to operate them and with greater uncertainty than we're accustomed to doing now. Did that work more or less?
Yeah.
Okay.
Amazing. Okay. I'm going to move questions to, we have drinks afterwards, is that right? Yeah, so I'm going to, please, going to give Kristala a break. Please join me in thanking her for an amazing talk and discussion. Thank you. Thank you. And before I invite you to join us for drinks, I also wanted to ask you to please thank amazing AV team and tech that allow us to record this. Everything has worked very well. Please thank the BSL interpreters as well. And I want to extend very special thanks to the IMSE team. They're somewhere around. Elena, have they just run out every year? Elena, Anne-Marie, Steph, and very special thanks to Leah, who is our events organizer. She's absolutely amazing, and she made this possible. She will be outside, so if you see her in the blue suit, thank her for this amazing day. And again, thank you, Kristala. Thank you all for being here. Join us for drinks.